Great. Okay, so I'm Nick Steinmetz. I'm an associate professor at the University of Washington, and I'm going to talk about cracking circuits with ultra probes, and I will introduce briefly the Opto probes, and we'll hear more about them from the subsequent two speakers. So in thinking about the roster of devices that Tim just told you about, there are some remaining challenges that we thought at the beginning of this project I'm going to tell you about we might be able to improve on. One is that the yield of sorted neurons is low. There's a sort of classic problem in extracellular electrophysiology, which is you stick a probe in a brain and we know something just from tissue histology about how dense the neuron should be, and yet we don't record as many neurons as we think we ought to given the density. So there are some various possible explanations for that in the literature. And one we thought was, well, maybe we just could be more sensitive and more discriminative with our probes. If we had a better recording device, maybe we'd pick up more of those neurons. Second, small neurons and processes are difficult to record. This is a cerebellar granule cell, which is famously difficult to record. I'm not going to come back to talk about whether you can record these with the device I'm going to tell you about, but I will tell you about some other small neurons and processes of neurons, like axons, that we made progress on trying to record. And finally, you have minimal cell type information in extracellular electrophysiology. So the plot here shows broad and narrow spiking waveforms, which is a sort of single classical feature that people use to discriminate putatively inhibitory from putatively excitatory neurons in the cerebral cortex of multiple species. And we thought, wouldn't it be great if we could record something in more detail that would allow us to have more than one bit of information about cell type, more than just broad, narrow waveform? Could we get more information about cell types? Okay, so to address those challenges, we designed a new version of the Neuropixels site layout. So I should say at the outset, to sort of clarify the connection with the past talks, this probe that I'm going to call Neuropixels Ultra is a 1.0 probe, but with a different recording site design. And so whereas the 1.0 proper probes have these 12 by 12 micron sites, and as Tim showed you, this sort of checkerboard arrangement, and the 2.0 sites have this vertically aligned, slightly closer arrangement, both of those for about 100 sites per millimeter in linear probe shank distance. The Ultra has 10 times that density. So there are 5 by 5 micron sites, and there's just 6 microns center to center in 8 columns. And so you get this super high density of recording sites. And as Tim mentioned, that density comes with a trade-off, because this is still just a 1.0 probe on the back end, which means that you still only read out 384 channels. So that gives a total coverage then span of 300 microns relative to the approximately 3 millimeters coverage along the shank for 2.0 and approximately 4 millimeters for 1.0. So that's a major trade-off. I'll talk more about that on the next slide. First, let me just acknowledge that this project, in terms of all of the data collection and analyses and so forth, was really led by Joanne, a postdoc in my lab, and Andrew Shelton, a really talented scientist at the Allen Institute for Neural Dynamics. And so, yeah, most of the work you'll see here is thanks to those two guys. Okay, so here is a schematic to give you a sense of the size. So this is not a photograph. This is a composite. I took a reconstruction from the Allen Institute database of an individual neuron morphology and superimposed it to scale over a Neuropixels Ultra, just so you can see how small and dense we're getting with these recording sites. And you can see how, this kind of density and this kind of small recording sites really gives the possibility of recording very small neurons. This is a big neuron type, but very small neurons, or even subcellular features. You can see that these recording sites are now on the scale of sort of axons and dendrites of neurons. And so indeed, when we put these in the brain and record individual neurons, we get these beautiful, highly detailed portraits of the action potential waveform of individual neurons. So here's 2 example neurons, and what I'm showing here is the average waveform recorded on each of the sites of the Neuropixels Ultra. So there's 8 columns in this figure. Each one corresponds to one of the columns of sites, and so every one of these little black traces is the waveform of one example neuron that was spike sorted and averaged across individual waveforms to give its average waveform on these different spatial locations. Okay, and maybe you see this colorful video, which is playing out the waveform over time behind it. It's probably not playing as smoothly over Zoom as I see it. But in any case, what you can see immediately is that, for some neurons, this density is hardly buying you anything. Like this neuron is about the same on a whole bunch of different channels. That's fine. For other neurons, you get quite a bit more detail in the spatial structure of the neuron. So this neuron has, a really different property over here than it does over here, and we can sort of infer, and I'll talk a bit more about how to interpret this later. We can infer that these are some different processes of the neurons, maybe soma on the side and dendrite on that side. Okay, so we talked about the span, and you may be looking at this here as well and saying, wow, we're just covering sort of 300 microns is like part of a single cell in the cerebral cortex of a mouse. Isn't this a problem? And so yes, it is. And I'll come back to what use cases this probe might be best for at the end. But first, let me just say, these probes are switchable. So in software, you can select different channel configurations to trade off the coverage and the density. So here I'm showing 3 different switching configurations that are possible. One of them switches from the original 8x48 grid into a 4-column by 96 row, and that grid, which gives you really high density down the middle of the probe and covers twice as much length. So that's now up to 0.6 millimeters. You can do a double column, which covers 1.15 millimeters, or even a single column of sites, 384 by 1, which then covers 2.3 millimeters. And that can be useful. We show in the paper something I'm not going to show you today, which is like recording back propagating action potentials at super high density along a long distance. So there are different channel configurations that you can select with these probes, so it's not fixed to that really small density. Okay, so if you know something about electrophysiology, you might be wondering at this point, like, what about the noise levels? Because it's a classic bit of knowledge in the field that if you make recording sites smaller, the trade-off is that the noise level goes up, and that's definitely a true property. So this plot was produced by Carolina, who you heard earlier, where the blue curve is showing exactly that result, that the electrode noise, the noise from the recording site, goes up as the recording site gets smaller over to the left here. And Neuropixels 1.0 is out here at 12 by 12 microns, 144 square microns. Neuropixels Ultra is down here at 25, 5 by 5. And so you can see that, yes, the noise of the electrode is higher than the noise of the 1.0 electrodes, but we're really not at the really steep part of that curve. And because the total system noise adds kind of sublinearly, that increase is actually pretty modest, just about 2 microvolts RMS. And we can see that reflected here in this measurements in saline. And moreover, the in vivo noise is really dominated anyway by the local neurons that surround the local neurons and neural activity that surrounds the place where you insert the probe. And so the typical in vivo noise is, at least 15 microns or more, often more. And so this couple of microns increase for Neuropixels Ultra is, not a big impediment, and really the signals look quite good and comparable to normal 1.0 recordings. Okay, so we wanted to understand the way in which Neuropixels Ultra might have advantages over 1.0 and 2.0. And to do that, we did this kind of analysis that's going to be the basis of the next slide's plots that quantify those advantages. And in this analysis, what we do is we take a recording that we actually made with an actual Neuropixels Ultra probe. So the actual recording had the full 8 columns, 48 rows geometry that I've been showing you. And then we subsample the data to get what the recording would have looked like if we had recorded with a 1.0 or a 2.0 like geometry at the same exact recording location. And so we can see for a given neuron, like this neuron here has a really high amplitude on these ultra sites, but because this neuron is really, really small in space, any of the 1.0 sites, none of the 1.0 sites see it with large amplitude. And so with 1.0, you get a really small amplitude. So this tells us what this neuron would have looked like if we had recorded it with a 1.0 probe. And using that kind of analysis, we can see that the amplitude of recorded neurons is much higher for Neuropixels Ultra than for 1.0 and 2.0. I won't explain this third condition. You can read the paper. And the signal to noise ratio ends up being higher. So basically this increase in amplitude outweighs the increase in noise that I just told you about and results in a larger SNR. The increase in amplitude, by the way, happens partly for this reason, which is that some neurons can fall in between the gaps of the other probe geometries. And partly because even when the peak is centered on the location of one of the sites, the gradient of the voltage is still very steep. And so having smaller sites just allows you to sample that peak more sharply. And so the net result is that SNR is higher, and the outcome is that the yield in terms of the number of units that you can record is about twice as high for Neuropixels Ultra than for 1.0 and 2.0. And this, of course, is a per unit area comparison. So this is over the 300 microns that are sampled by Ultra. You can get about twice as many units over that small range relative to 1.0 and 2.0. So of course, I want to be clear, like if you're doing a calculation that imagines that you have a brain region that's 4 millimeters long, and you're happy to have neurons anywhere within that 4 millimeters, then of course the total yield across that 4 millimeters is going to be much higher for 1.0, where you can have sites across that whole 4 millimeters. But if you have a brain region that you care about, which is only 300 microns long or fits within that 300 microns, then this probe would maximize the yield for that smaller region. Hopefully that makes sense. I'm happy to answer questions about that. Okay, so now I'm going to tell you about something curious that we observed when recording with these probes that led us to some biological insight, biophysical insight, let's say, that I think is useful for people in the field to know about. So here is a spatial map of one neuron with the waveform in white and a sort of like interpolated spatial profile of that waveform in the color map behind. And you can see, again, this is an example of a neuron like the one we saw a couple slides ago, where the spatial footprint, we'll call this the footprint, is really small. That is the extent on the probe. So from the peak site to just 2 sites over, which is just 12 microns over, you go from a high amplitude waveform to nothing. So this is really small and spatially restricted. It's not, for instance, like the thalamic example neuron we saw before. And we recorded all over the brain in the mouse from a bunch of different structures, and we saw these neurons in lots of different places. So here is an example of a large waveform neuron from visual cortex and a small waveform neuron, a small footprint waveform from visual cortex. And here's the distribution of all the footprints. And you can see that there's a sort of a second peak of these small footprint waveforms. And so they appeared initially just to be a separate class in some way of waveform. And I'll emphasize actually that the good news, well, let me emphasize that in a sec. So we recorded from a bunch of other areas, and we see a similar thing. We find these small footprint waveforms in lots of different regions. And in many cases, it does indeed appear to be a similar sort of second peak or bimodality of these small footprint waveforms. Here's a quantification of the percentage of units that have these small footprints over a bunch of different regions that we recorded from. And so it's fairly constant, about 10% of units in most regions that have this small footprint property. And I'll emphasize that this is broadly good news for the Neuropixels 1.0 and 2.0, because that means 90% of the neurons have these large footprints, which are quite reasonably well sampled by the 1.0 and 2.0 less dense geometry. So that's broadly good news. Okay, but what are these? Oh, so one more slide. So we also included recordings from multiple different species, and we find the same observation in those different species. You can find these small footprint waveforms at a similar percentage of the total number of neurons recorded in different species. So what are they? We kicked around this question from different perspectives for a while, and finally we hit on the real ground truth experiment to prove that they are in fact axonal recordings. And the experiment here, just to let you know what the evidence is here, we apply mucimol to the brain. Mucimol is a GABA agonist. It activates GABA receptors. And so anything with GABA receptors is going to be turned off by the application of mucimol. And somas and dendrites all have GABA receptors, but axons don't. So if you have an axon coming in from some distant brain region, it doesn't have any GABA receptors. And so any activity on that axon will be unaffected by the mucimol application. And so here's what happens when we apply mucimol to visual cortex in the mouse. We apply mucimol here, and you can see most of the neurons shut down, but a few of the neurons proceed to continue spiking. And when we look at which neurons are the ones that continue spiking, that is, which ones are resistant to the mucimol application, it's all of those resistant neurons are small footprint waveforms. So it's not the case, just to clarify, it's not the case that all small footprint waveforms are resistant. This could, of course, be some, these small footprint ones that are sensitive could be axons from local neurons, which have their local neurons themselves turned off. But in any case, what we can claim is that all resistant neurons are small footprint, which is all axons have small footprints. Did I say that right? In any case, so here's the main point that I want to make, which is the small footprint. Here's two examples of small footprints. The small footprint in our hands was a more diagnostic feature of being axonal because it has this really, really strong relationship with the mucimol sensitivity than this particular waveform shape itself was. So here's two different examples, one of which has its upward going waveform shape and one of which has a downward going waveform shape. And both of these were mucimol resistant, which is to say these are definitely axonal recordings. And so this is something I think it's important for everyone who's doing electrophysiology, not just with these Neuropixels Ultra probes, to be aware of, which is that sort of a classic concept was that upward going spikes might be axonal. The logic was that if the spike is generated somewhere far away and sort of propagating towards you, then you get this upward deflection in the waveform as the spike is on its way, and then a downward one as it arrives to the part of the axon that's near the recording site. But we see these units like this, which are definitely axonal, but which have purely downward or nearly purely downward waveforms. Their diagnostic feature is that they're small footprint. And in some computational modeling that we show in the paper, we believe these are recordings from nodes of Ranvier, or at least, let's say, very likely to be recordings from nodes of Ranvier. They're consistent with what a computational simulation shows from a recording of a node of Ranvier. So I think if you don't want to record from axons, and these are not only axons, they're long range axons because they weren't affected by the mucimol application. If you don't want to be recording from those, then you need to consider that footprint is a feature that you should calculate for your waveforms and exclude the ones that have small footprint. So to summarize, on Neuropixels Ultra here, they have much greater site density while sacrificing span. This density confers higher yield, better detection of these small footprint waveforms. I didn't really show it, but obviously these small footprint waveforms are detected better on the high density of Neuropixels Ultra. And better cell type discrimination, which is the last figure in the paper, which I decided not to go over in the interest of time, but you can read the paper to check out what we showed there in terms of discriminating between PV, VIP, and SST interneuron types within visual cortex. And so with Neuropixels Ultra, we discovered that footprint is a useful waveform feature, particularly for identifying axons. And so who are these probes ideal for? I think they're ideal for targeting small structures. If your brain region of interest, if the thing that you want neurons from is about 0.5 millimeter or less, then I think you can do probably better in terms of identifying characteristics of the neurons you record and better in terms of the yield of your single units from a Neuropixels Ultra probe than from a traditional 1.0 or 2.0 probe. Of course, many of us, my lab included, are primarily these days interested in recording multiple different structures at the same time or recording really large areas within a structure. And so for that, I absolutely believe that 1.0 and 2.0, of course, are the better probes for that. And we use those dominantly in my lab for scientific applications because of those considerations. But again, like if you're interested in targeting just one particular thalamic nucleus, say, then this could really be the probe for you. They are available for purchase now, as Tim mentioned. They're called 1.0 HD, so that's high density, and the 1.0 referring to the fact that they are, as I mentioned, a 1.0 probe on the back end. All right, so let me just briefly, in one slide, introduce Neuropixels Opto, which is what you're going to hear about in the next two talks. And basically, Neuropixels Opto says, okay, if you really want cell type specificity to a much greater extent than what we're able to achieve with the Neuropixels Ultra, then the way you're going to have to do it, especially in terms of genetically defined cell types, is with accessing them via light. And in addition, being able to deliver light into the brain allows you to do optogenetic manipulations in a localized way. So those are the two topics Carolina and Anna are going to tell you about. And so what this Neuropixels Ultra probe does is has light emission sites every 100 microns along the shank. And they're in two colors, red and blue, and they come down via waveguides via a laser that's coupled into the probe. So this is what's called pre-alpha version of this probe. In fact, I think this is like the pre-alpha version of the probe that we're showing here. So early prototype, the early prototype here has this laser fiber coupling in and bringing light down the waveguides to emit at the different sites. But as you'll see in the data from Carolina and Anna, these probes nevertheless have great performance and allowing us to record both the electrical activity at a 2.0 like site geometry alongside being able to do this local simulation for manipulations and opto tagging. So yeah, looking forward to hearing Carolina and Anna tell you more about that. So let me just wrap up by thanking the Neuropixels Ultra team. Again, Joanne and Andrew were the leaders of this project with a great group of collaborators. And these are our funding sources. Happy to take questions.